Great, so I'll let people in.
Perfect. Great. We're going to let people join. Okay. Okay. So I'm going to start. We're at the top of the hour. Welcome, everyone, to the very first episode of ALS Canada's Clinical Trials Unboxed. Hopefully, we can get a theme song for this eventually. This is going to be a every other month webinar, a very short one, where we can highlight research opportunities in ALS and clinical development in ALS. We'll put at the outset that we are not endorsing any particular company or experimental therapeutic, but we do endorse research opportunities. And for both Canadians and anyone who may see this who is out there outside of Canada who wants to participate and help move the needle towards better treatments for this disease. So this is very exciting. For the first episode, we have a special guest. Our Chief Medical Officer of Verge Genomics, Dr. Diego Cadavid. And you may have heard of Verge Genomics back around 2015. Their CEO, Alice Zhang, was a scientist who decided she wanted to put together a company for drug discovery using artificial intelligence, big data, and using unique human-based cell models to start doing this drug discovery. We heard around 2019 that they had some interesting therapeutic targets, and we've recently learned in the past few years that one of the first ones they're moving forward through VRG50635, which you'll hear about today, is around a particular target called PIK5 kinases. And there is some interesting preclinical work, mostly by Dr. Justin Achida in California. I'll put in the chat a couple of the publications so you can check out PIK5 and its relation to ALS while the talk, not while the talk is going, but for afterwards. And that's about it for now. Really excited. We'll take some questions and answers after. And if you have other questions, you can certainly insert them into whatever social media you might be watching this on later. So without further ado, I pass it over to Diego. Thanks so much. And the floor is yours.
Thank you, David. And good afternoon, everyone. I will give you an overview about our clinical program of a candidate that we are studying as a potential treatment for ALS. It's a small molecule, which means it's just a pill that people take by mouth. I'll refer to it as 635. I'm the Chief Medical Officer. Verge is a company that's about 8 years old, based in the San Francisco Bay Area. So first, an overview of what I will be sharing with you. This small molecule 635 was designed for a novel drug target. It's an enzyme called PIK5 that plays a really important role in the traffic of proteins and molecules inside cells. This target was identified as a promising target by people at Verge working in tissues from people who had died from ALS. And also it was independently identified by Dr. Ichida doing phenotypic screens. We have, and Dr. Ichida independently have a lot of different efficacy in cells, in animal models. I will be sharing some of this with you. 635 has been studied and has actually moved into people. I will share that data with you. It has what we call favorable drug properties. When people take it through the mouth, it gets to the blood, it gets to the spinal fluid. We have shown that it gets to the brain and the spinal cord in mice and rats. And we have done already initial studies in healthy volunteers. More than 100 humans have taken this compound. So with this data in hand, we recently initiated a study in people with ALS. And David was very kind to invite me. Our first site was actually activated in Canada. We have other sites coming in Europe. I will share with you a bit of the timelines. Clinical trials are a long process. They're very complex. I will share with you how we are thinking that drugs can be developed for ALS, so we acquire more data faster and we learn earlier what drugs actually look promising. So we stop working with the ones that don't look promising and we favor the ones that look good. And the idea is that once we make a decision to go to a phase 3 registrational trial, we really understand how this drug works, what dose should be used, what safety and tolerability is expected, and that we have high probability of success. So this is a cartoon that shows something that took Verge, scientists at Verge over 4 years. First was collecting tissues from people who have died from ALS, both spinal cords and brains, but also control tissues from people who have died from other diseases or other causes. And some of these tissues were sequenced by Verge, others were sequenced by academics. Altogether, 988 tissues from 983 human beings were analyzed using very sophisticated analysis for RNA transcripts. And this generates a map of what genes are turned on or off in tissues from ALS versus tissues from people who don't have ALS. And this we call the data atlas. Through a lot of machine learning, artificial intelligence, a process that took many years. It's really not doable by one person because there is so much data and you need to understand how all this data is moving together, what's connected, what's not. Through this long process, Verge ended up identifying one signature that we think is the key impairment in people with ALS depicted in the second figure. Something that's really exciting is that several of the genes that are known to cause ALS are actually part of this network. So that gives us high confidence that this network is involved in ALS, both through genetics, but also through our analysis of tissues from deceased people. And then the next phase on the third figure is how can we try to restore this network so that it functions better, ideally normal. So again, a lot of data analysis, a lot of artificial intelligence. We ended up identifying one drug target, a kinase. Kinase means a protein that phosphorylates other lipids and other proteins as a key target to try to restore the network by inhibiting it. So Verge has discovered PIC5. Dr. Ishida independently also find it doing a very different approach. And now we have developed a small molecule, which is a PIC5 inhibitor. And that's what we're testing in the clinic. I'm gonna skip this slide to make sure I have enough time, but I wanna share with you, we've done extensive testing in cells. Cells, for example, skin biopsies from people with ALS can be used to generate motor neurons. And when we incubate these motor neurons with mutations that cause ALS, with this compound, we can restore their survival back to normal. So this is one of the assays that we use that give us a confidence that this small molecule, a PIC5 inhibitor, has the potential to protect cells that come from people with ALS. Dr. Ishida published last year a comprehensive study. I think David will share it. It's obviously highly technical, but what's important is that his team tested cells from people with sporadic ALS, meaning they don't have known mutations. And all of them seem to have improved neuronal cell survival when incubated with PIK5 inhibitors. He also tested different cells from people with different mutations that cause ALS, and again saw this beneficial effect on cell survival. And also when PIK5 inhibition was put into mice, either genetically or through pharmacology, again, there was a benefit in motor function and a benefit in survival. So all of this together gives us a reason to test PIK5 inhibition in people with ALS with the hope that we could result in cell survival, motor function improvement, and find an effective treatment for this really terrible disease. I'm going to show you, this is a video published by Dr. Ishida of 2 mice. One of them was treated with a PIK5 inhibitor, and the other was given a control, a not specific control. And these mice are engineered to express a protein called TDP-43, which is believed linked to the cause of ALS, and it's found in most people who die with ALS. So think of this as mice that are very sick from too much TDP-43. So you can see, although both mice are sick, one of them is functioning much better. And that's the mouse that was given a PIK5 inhibitor. So right now we have what we call a clinical development plan at Verge. We have designed a series of studies, and this is an outline of all the studies. We began by testing healthy volunteers in 2022, and a large portion of 2023. We have learned about the basic properties of the compound, the initial safety and tolerability. We have measured the levels in the spinal fluid. We have confirmed that it has pharmacology in the spinal fluid. All these are foundational studies that need to be confirmed before we advance. This year, as you can see, we are we're in the middle of a second study in healthy volunteers, where we are testing a new formulation of the compound, a more advanced, better manufactured compound, a formulation that can be used through feeding tubes for people who cannot swallow safely. But we're also launching a first study in people with ALS, which you see here, study 002. This study actually started in Canada in December. The first patient has been enrolled, and we are about to enroll additional patients. Also, we are about to activate 7 sites in Europe, and we plan to have about 50 patients enrolled and completing about 6 months of treatment about a year from now. And I'll show you more, but we believe all that information will actually tell us if PIK5 inhibition with our compound can actually treat ALS. That's not enough to get approval. To get approval, as you see, we need to get longer safety data, and we need to do additional studies comparing the drug to a dummy pill called a placebo. And we are planning to launch 2 studies, one that is smaller and faster using biomarkers. Some regulatory agencies may be willing to accept an earlier filing if the data with biomarkers looks very good. Other regulatory agencies are not willing to do that and would need to wait for a more classic study using disability scales, and that's the phase 3 study that you see there. So this gives you us a sense why drug development takes so many years. So this is just rapidly an example of what we did in healthy volunteers. We completed this study. We began by giving single doses, small doses to groups of 6 or 8 people or placebo. And if it's safe, we increase the dose. You can see we went from 60 to 180 milligrams. When we get to 540 milligrams, then we started whether taking it with food impacts the absorption of the drug. So we did it fasting, high fat, normal fat. We concluded that normal fat works best, and we increased the dose to 1,000, 1,600, and then we enroll more groups of healthy volunteers, tested 200 milligrams once a day for 7 days, then 400 milligrams once a day for 14 days, and then 1,200 milligrams. We did spinal taps, lots of safety work. So that just gives you a sense of everything that we do to make sure we understand the drug effect, not only in animals, but also in humans. And now we are actually initiating this ALS study. This is a cartoon of the study. This study begins with what we call a pre-treatment run-in. So when patients are enrolled for the first 8 weeks, we are measuring everything, their blood, how they move, how they breathe, their muscle strength, how they're feeling. I'll show you in a minute. We are deploying a series of really cool measurement tools to learn with precision how every patient is functioning before we give them the drug. Then they move into the drug treatment period, where we're gonna be testing increasing doses of the drug. Each of these dose levels has the potential to be efficacious, and they're all in a range that is considered safe. So as we increase the dose, they're gonna be at 8 weeks at each dose level. We're gonna be learning the safety, the tolerability, and whether any of the measurements that we took in the pre-treatment period is changing. Markers in the blood, how they're walking, how fast they're walking, how they're using their body. So we do that at 3 increasing dose levels. And then at week 40, we're gonna take an interim look when we have 40 patients treated for 40 weeks. And at that point, we will make a decision. Do we see that this treatment is having efficacy in ALS? Is it being tolerated? Is it safe? And then patients, of course, continue on a long-term treatment for another 40 weeks. So that's the essential study design. There is no placebo. The comparison is to the pre-treatment running period and to what we call digital twins. We already have a lot of data from other people with ALS, and we understand the natural progression of the disease. One of these exciting markers of ALS, it's a protein called neurofilament light. This is a protein that comes from the axons. Axons are the long extensions of neurons. The motor axons are very long. Imagine they have to reach from the brain all the way down to the spinal cord, and then from the spinal cord to your hands and feet. These axons are very long. ALS kills the neurons, and when the neurons die, these axons degenerate. So this protein leaks into the blood, and we can measure it in the blood. The amount of this protein in blood is a very strong predictor of function and survival. And we believe it's also a predictor of a treatment effect. So if our drug is working well and increasing the dose gives us more efficacy, you can see in this cartoon, as the patients run through the pre-treatment, dose 1, dose 2, dose 3, we will see a progressive decrease in neurofilament light in plasma. hopefully to a plateau where very few neurons are dying. These are also some of the really innovative tools that we are using in the study to measure the function of the patients. We are measuring speech. The patients are giving a tablet and they connect on the web to an avatar, a woman called Tina, and they have a conversation with Tina. And we use artificial intelligence and algorithms to measure the changes in speech. Very, very detailed assessment. We're doing this every 2 weeks. We're also measuring the breathing because ALS particularly affects the muscles of breathing, how much air they can move. And again, we're doing this every 2 weeks, sending nurses to their homes or having the patients go to the clinic. We're also giving patients accelerometers. They're like a watch that they wear on their wrist and in their waist 1 week every month. So that when they're moving around in the home or outside the home, we can measure the quality of their, what they call the sub movements. Every time the arm is creating a movement, the accelerometer will record that. Very important for fine motor movements. And also really cool, we are setting up the homes with something called the Emerald device. This is technology out of MIT. It's like your Wi-Fi in your home. So it's very private, it's very safe. But these devices basically are like radar. Whenever you move in the home, this device will be able to measure that movement, not only when the patients are awake, but also when the patients are sleeping. And we're doing that 24/7. And it's analyzed by, again, artificial intelligence, machine learning, because it's so much data no human can analyze. So we're gonna learn a lot about both the disease and what happens when patients are taking increasing doses. This is an example of a living room. You can see the Emerald sensor on the wall. And because we collect so much data, the table on the low part of the slide tells you that we can detect changes in gait speed that are very tiny with very high confidence. So we will know early on if this drug is working or not. And right now, I just want to finish, share with you the way drugs are approved for ALS these days is using a scale called the ALS-FRS-R, which you see on the top. This scale is very old. It's 25 years old. It's based on asking questions to the patients about your swallowing, what we call bulbar function, your breathing, and also your fine motors and your gross motors. So there are 12, essentially 12 functions being assessed, grouped into 4 domains, and they're all integrated. This is a very gross measure. It's not a very good tool. Regulators like it because it's so gross that if a drug moves it, they feel very confident the drug is working. But it's a very bad tool early in development because early in development, we don't have hundreds of patients enrolling to each treatment arm. So we need much better measurements early on. And at Verge, I'm very confident we are putting the right measurements to tell us whether our drug is working or not before we move into larger trials that will use this very crude measure called the ALS-FRS. And with that, I just want to finish by sharing a bit of a story. I've been doing drug development for over 20 years. It's a team sport. It's a very hard team sport. Everybody has to work like David and Colleen are doing as a patient advocacy organization. As some of you who one day may choose to join clinical trials, your doctors who choose to participate as sites in clinical trials, companies like Verge that are willing to bet that one day we will have safe and effective treatments for ALS. The regulatory bodies that make sure we do it right, and that only drugs that are developed ethically and that are safe and effective are approved. The funders of research, the people who give their capital to Verge, the governments, and philanthropy. And if we have time, I'm happy with David's help to answer a few questions. Thank you.
Great. Thank you very much, Diego. So we probably won't have time for any of the questions in the chat, but please feel free to put them in there if you do have any, or if this is something you're watching afterwards, please feel free to put a question underneath the video and we will get back to you. I think one of the questions we had that will likely come in a lot will be around Canada and the potential for Verge to have either more sites in this particular trial, or as a drug discovery company, presumably there may be more ALS targets in the future. Is there a good chance that there will be other Canadian sites that Verge might connect with either later for the 635 or with other potential experimental therapeutics?
Yeah, thank you, David. We have 2 sites in Canada out of 9 for this study, which is enrolling 50 patients. If this study shows positive results, as I show you in the clinical plan, we will open a second cohort, and we plan to add more sites in Canada. We will share all that information with David and Colleen. If we go to phase 3, we will open even more sites. I can tell you there are many companies working, trying to develop drugs for ALS. It's an exciting time. It's a very difficult disease to treat, but this is a time where we all should join forces and work together and make it happen.
Great. Well, thank you for that. That's great news. I'm sure people will love to hear that, and we'll certainly inform people of where those sites are as they come along. And we really look forward to, actually, from your last message, we really look forward to taking this innovative design that you've got and this pathway through to a pivotal trial, and working with Verge as you work with the whole field to sort of ingratiate them to this way that you're doing the trials, and how the communications come out afterwards if things are looking positive, and moving into a more pivotal trial. So that's very exciting, and looking forward to watching that move forward. One of the things I think you'd mentioned too is that it was a biomarker driven trial. Are there going to be target engagement biomarkers to show that you're hitting the PIK5 kinase?
Yes, this is something we've already done in the healthy volunteers, and we will use that in the people with ALS too, David. Very good point.
Perfect. That's great, because no matter what, then science can evolve if we get those kinds of answers. There is one question in the chat, which is, where can we access the recording? This should end up actually on our social media channels and on ALS Canada's YouTube. So we will be pointing people through our different channels to that as this one goes up, and then as we have more of them. Kevin Robinson actually asks about sites in the US. And whether or not you've applied to the CDMRP at the DoD for any funding.
Yes, so we plan to open sites in the US if we move to cohort 2. Right now we're working in Europe and Canada. And in terms of applying for funding, no, we have not, but we are aware that some of our peer companies have done that and have obtained some funding, but we have not.
Great. Well, we're actually at the end of the half hour, and we were very committed. We're going to keep these to half an hour, but maybe there will be even a chance we can have you back when things are further along, and we can hear about some of the results and have a conversation about those in another format. So very much appreciated for you to join us and Verge to join us for this very first one. Really exciting design, and we look forward to seeing how it moves. And thank you to everyone who has joined today. And I'll just add, Kevin Robinson says he would encourage you to apply to the CDMRP. So I'll just add that in there for extra funding. It's definitely a great pool of funds for translational research. So if not even for the 635, I'm sure there's a lot of other potential candidates that might be something for. So thank you everyone for joining. Looking forward to seeing you again in another Clinical Trials Unboxed, and have a great day.
Thank you, everyone.
Nice.
Thanks.